Colette.
And I'm Sarah.
I'm Shelbs.
I'm Sidge.
The purpose of this lab is to silence the gene that triggers mutations in C. elegans. So we did this by introducing the RNAi system that was in the bacteria that was fed to the C. elegans. The big picture purpose is also to work with modeled organisms because of their similarity with human genes.
The different method we used included chunking. Chunking began by rubbing your hands with hand sanitizer and then putting on gloves, goggles, and tying your hair back. You also had to roll up your sleeves past your elbow. The biological safety cabinet needs to be sterilized. To do this, we spray down your arms with rubbing alcohol. Place the spatula in the bottle full of alcohol. Light the flame and run the spatula through the flame twice to ensure that no condensation is on the spatula.
Move starter petri dish with one finger and remove the top of the lid, hovering over the dish, not removing the lid completely off. Then use a spatula to cut a small square and scoop it onto the spatula. While holding the spatula in one hand, close the lid and move the starter plate with the finger away from you. Open the feeder plate with the lid, hovering over it again, and gently place the bacteria on the outside of the petri dish. Close the dish and slide it away with one finger. Run the spatula through the lit flame one last time to remove any bacteria.
Wild-type worms were present in all life stages. The majority of them were skinny, while the eggs and lower stages of life were covered by the higher stages of life. An egg is the lowest stage of life, as L1, L2, L3, and L4 follow. The last stage of life is adults. These are noticeable with eggs present inside of them. They moved at a constant pace in a sinusoidal pattern. The blisters were hard to identify on most C. elegans, yet we were able to find a couple.
The rollies were very easy to notice, as in vast majority they were moving in circles. The seizures were small movements of shock that left the worms in a straight line, leaving them to die. Lastly, the dumpies were all short and wide.
When observing wild-type C. elegans, after placing them in the OPO50 bacteria, the life stages that were mainly present included L1 and L2s.
With the dumpy C. elegans, there were mainly L2 or L3.
The blister worms were mainly deceased adults.
And the rollies were present in L1 or L2 life stages. Although we did not have any present worms moving in circular motions, we looked at Miss Petrie's plate.
The BLI1 gene produces proteins called collagens, which is a structural protein found in skin. It is a mutation that disrupts the structure of an adult stage worm, creating fluid-filled blisters.
The ROL6 gene causes the C. elegans to have the right roller phenotype. They are twisted into a right-handed helix. They encode for the protein called collagens.
The dumpy gene encodes a membrane associated with the thyrodoxin-like protein.
Our blister plate worked extremely well, and almost 100% of the worms were full of blisters. A lot were dead because of the severity of the blisters that occupied the entire body of the worm. The gene that was silenced caused the adults to form these blisters and die rapidly, as the blisters were still present after they were deceased.
There were no present rollers in our plate. This could be due to contamination when transporting the worms to the feeder plate. Another error would have been overpopulation, where the chunk of C. elegans would have contained mostly adults or L4 stages and eaten all the bacteria.
The majority of the worms were not all dumpy, but the ones that were clearly distinguishable.
The feeder plate contained the double-stranded RNA that would be eaten by the wild-type C. elegans. The DS RNA enters the cell and triggers the RNAi pathway. The dicer breaks up the DS RNA into short strands of RNA. These new short chunks are called siRNA. They then bind to proteins called RISC. The double-stranded siRNA are unzipped, leaving the single strand of RNA. RISC binds to its matching mRNA, then splits it, silencing its function, as the split mRNA can no longer be translated into a protein. Then the mutation occurs because of the silencing of the specific gene that prevents the protein from being made, therefore the protein is not able to be expressed.